Hi, this is Mrs. Bustamante. Today we're going to talk about the cellular respiration diagram. And I know this diagram looks really complicated. It is probably because it is. But I'm going to try and use this diagram to help us put together the three pieces we've learned about with cellular respiration to get a better idea of where they're happening in the cell and how and why they all relate to each other. So I'm probably going to break this video up into a couple of parts. We're going to talk about our overall picture and probably the first part of cellular respiration in this first video, and then move on to the Krebs cycle and the electron transport chain in another video. So first, what I want to point out is we're going to talk about the process of cellular respiration, and you should know the overall equation for cellular respiration. It's that we start with glucose, C6H12O6, and we add 6 oxygens, and then we get out 6 carbon dioxides, 6 molecules of water, and up to 38 molecules of ATP. So this is our overall equation for photosynthesis, or cellular respiration, and you should know that. You should also know that it's the opposite of photosynthesis, right? It's the reverse of what we saw in photosynthesis. It's why they pair together. It's why we talk about in photosynthesis, you make glucose, and in other organisms that aren't autotrophs, they ingest their food somehow and they make glucose from those molecules in their food. So these orange boxes you'll see on my diagram, these orange boxes refer to the reactants. So we're going to trace where are the major reactants happening in these processes as we move through the process of cellular respiration, right? Where do they come into play? And then where do we make these products? And we're going to draw our products in green. So you can do this as you go along, or you can pause the video a lot, or you can fill everything in and then listen, kind of what works best for you at this point. But I am going to go fairly quickly, probably too quick for you to write and color at the same time. So you will have to come up with kind of a plan for yourself to help you accomplish all this. Okay, so we talked about our overall cellular respiration equation. I want to talk about the first step of cellular respiration. The first major process of cellular respiration we have is called glycolysis. And I'm going to put our big processes in red. And this first process is glycolysis. It's happening in the cytoplasm of the cell. And it's important to know that it's happening there. And the process of glycolysis takes glucose. Here's glucose written as a word, and its formula is C6H12O6. It's a 6-carbon molecule. I know that because of this 6 here. And we take that glucose molecule and we have to add some ATP. And remember, ATP is the energy source of the cell. It's really usable energy. And we take that 1 molecule of glucose and we turn it into 2 molecules of pyruvic acid. And the reason we turn it into 2 molecules is that pyruvic acid is a 3-carbon molecule. So we took our 6-carbon molecule and we basically split it in half. So now we have 2 3-carbon molecules, which still helps us add up to 6. In order to do that, we input 2 ATP molecules. So we have to use a little energy, but we're going to see that we get out 4 ATP molecules from the process of glycolysis. We're also going to get out a high energy molecule called NADH, and we're going to get 2 of them, and we're going to see that used later on. Because we get out 4 ATP from glycolysis, but we have to input 2 ATP, we say that we get a net of 2 ATP from the process of glycolysis. You'll notice these ATP are in green. That's because eventually we're going to use them to add into our 38 ATP that we see in our overall equation. So if I were to say, what's the main point of glycolysis? Well, the main point of glycolysis is to take glucose and to turn it into pyruvic acid. And the reason we need to turn it into pyruvic acid is we're going to use it in the Krebs cycle. We get out a little bit of ATP from glycolysis, which is important, and we get out a high energy molecule of NADH. We get 2 of them. So that's kind of our goal from glycolysis, and we're going to see how these molecules move on to other parts of the cell in just a little bit.